Hello and welcome to Datta Biology classes. Today our discussion will be on DNA, deoxyribonucleic acid. DNA, RNA, these are not well told to you. Just a slight recapitulation before starting. DNA is found in cell. Each and every cell has DNA. Within the cell there is nucleus, and with the nucleus there is chromosome, and within the chromosome the DNA or genes are situated. But DNA is also found in chloroplast. And in mitochondria also. DNA is found in chloroplast as well as in mitochondria, or in each and every cell in the chromosome DNA are present. DNA is known as deoxyribonucleic acid. And what is RNA? RNA is ribonucleic acid. Clear? RNAs are formed within the nucleus of a cell. Nucleus of a cell contains chromosome. In the chromosome, RNAs are produced and they come out of the nucleus to cytoplasm. There are three types of RNA. We know that in the cytoplasm there is ribosome. Within the ribosome, the RNA is found known as ribosomal RNA. And in the nucleus, by taking some information or message or intimation, RNA is produced. Those RNA are known as messenger RNA. Clear? RNA found in ribosome are called ribosomal RNA, and the RNA which carries the messages from DNA of the nucleus to the cytoplasm or to the ribosome are called messenger RNA, which they act as messenger. And another type of RNA are available here. They are very small. They remain scattered in the cytoplasm. Those are called tRNA. These are the three types of RNA we will discuss later. Now, our discussion will be on DNA, deoxyribonucleic acid. Now, let us think that why it is called deoxyribonucleic acid. The question comes, why it is deoxyribonucleic acid? Ribose means C6 is hexose, C5 is ribose. So ribose is a sugar. It is ribose sugar, but it is called deoxyribose sugar. Why? Let us draw the structure of ribose first. If question is asked why it is deoxyribose and why RNA sugar are called ribose sugar, first we learn what is that. This is a 5 carbon sugar. This is C1, carbon 2. So this is carbon 1, carbon 2, carbon 3, carbon 4, carbon 5. Carbon 4 is here. Carbon 5 is here. So it is C5. this is H. This is H. This is C5H 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. H10. So 1, 2, 3, 4, and this one is what? 5. 4, 5. This is known as ribose sugar. Ribose sugar is a 5 carbon sugar. What is absent? Deoxy. Deoxy means removing of oxygen. In case of deoxyribose, this O is absent. So C5H10O4. This is known as deoxyribose. Clear? So if question is asked, what is the difference between DNA and RNA? You will say in deoxyribose, one oxygen atom is absent. You will get half marks. But if you write that in the deoxyribose sugar, one oxygen atom of the C2 carbon is missing, you will get the full marks. Because it is from the second carbon position the oxygen is missing, not from ours. So this is the difference. So deoxy means dehydroxy, removal of hydrogen. Dehydrogenation, removal of hydrogen. Deoxy, that is oxygen is removed. So in deoxyribose sugar, the oxygen atom of carbon 2, second carbon is absent. So it is called nucleic acid. It is available in the nucleus of a cell. In the nucleus of a cell, it is nucleic. And acid, some phosphate group remains attached. Phosphate group is acidic in nature because it has the ability to donate H plus ion. That's why it qualifies to be an acid. So it is a deoxyribonucleic acid. Now, we go into the detail of this deoxyribose. There are nitrogenous bases. It is known to everybody that nitrogenous bases are of two types, purine and pyrimidine. Or you can say purine and pyrimidine. This purine nitrogenous bases is double ring structure. No need to go into further details because we are discussing this. Purine has two bases. Both are double ring structure, and pyrimidine are single ring structure. These two are pyrimidine, these two are purine. Purine bases are known as adenine, guanine, and these two are known as thymine and cytosine. This is thymine, not thiamine. This is known as thymine. 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 So adenine, guanine, thymine, cytosine are the 1, 2, 3, 4 types of bases. These bases, nitrogen containing bases, the purine bases are double ring, while the pyrimidine bases are single ring structure. They remain attached with this sugar. With this sugar, once purine is attached, Another purine should not be attached there. Once it is attached with pyrimidine, it cannot be another pyrimidine with that. How? Let us discuss that. Say adenine is this adenine. This is known as adenine. This is double ring structure. These adenine nitrogen bases remain attached with this sugar. With this sugar. This is a sugar. One, two, three, four, five. C1, carbon 2, carbon 3, carbon 4, and carbon 5. C5 here. So this with this sugar, deoxyribose sugar, it is deoxyribose sugar which is absent here. Anyway, it remains joined. This adenine base, this adenine remains joined at C1 carbon. Very, very important question. Where the bases remain joined with the deoxyribose sugar, we know that it remains joined in carbon 1 position. Okay. Then, first, whenever it is remaining alone, it is known as adenine. adenine. When it remains joined with sugar, it is known as adenosine. Adenosine. And these two are collectively called nucleoside. Now, these pyrimidine bases or purine bases or whatever it is, these bases remain joined with sugar at C1 position, and when it joins, it becomes nucleoside. And then there comes phosphate. There comes phosphate. The phosphate remains joined here in C5. Phosphate remains joined. So it becomes nucleo. This one, phosphate, sugar, and nitrogenous bases. The three are collectively called nucleotide, nucleotide. So it is known as deoxynucleotide. More precisely, deoxyadenine nucleotide or deoxyadenosine nucleotide. So there are four types of nucleotides based on the presence of these bases. This is known as deoxyadenine acid. This is acid. When it remains joined with guanine, deoxyguanilic acid. When it remains joined in thymine, deoxythymidinic acid. And when cytosine is known as deoxycytidinic acid. There are four types of nucleotides. So these four types of nucleotides are called tetranucleotides. How? This is one adenine. Let me go to another one, adenine. Guanine. This is guanine. joins with sugar. This sugar is like this. C1, C2. So this will remain joined with sugar at which position? C1 position. Now, this is known as deoxyadenilic acid, deoxyguanilic acid. Then another thymine. Thymine is single. Thymine is joined with this sugar like this. C1, C2. So this will remain joined with this. And last, it will remain with cytosine. Join here, O, C1, C2, C5. So this is deoxyadenilic acid, deoxyguanilic acid, deoxythymidinic, deoxycytidinic acid. Now, these four remain joined here with C3 and C5 and H3PO4. In C3 we have seen some OH is there. OH and C5 is known as CH2 OH. This OH and then with PO4. This will be joining this nucleotide with this nucleotide. Again C3, this is OH and this is known as CH2 5 carbon, CH2 OH. So this OH, PO4. Phosphate bond, this is known as phosphodiester bond and coming. These bonds are known as which bond? The nucleotide remains joined with one another in the sugar in the C3 position and C5 position. It remains joined with phosphate. This bond is known as phosphodiester bond. Phosphodiester bond. This is the backbone. Remains joined, that is one nucleotide to another nucleotide to another nucleotide to another nucleotide. They remain joined by phosphodiester bond. And what is the name of this bond? This bond, this bond, this bond, this bond. Question may come, say beta glycosidic bond. This is known as beta glycosidic bond. So the nitrogenous bases remain joined with deoxyribose sugar at C1 carbon with the help of beta glycosidic bond, and the nucleotide joins remains with another nucleotide by phosphodiester bond. Clear? And these four are collectively called tetranucleotide. Let me clear it more perfectly. Suppose this is sweet this sweet ball joins the adenine at C1 position. You will never forget that in her left hand she is holding a balloon, that is nitrogenous base with the help of glycosidic bond. And at C5, if we put phosphate here, this is known as adenosine. Now, if I put here one phosphate group, it's known as adenosine monophosphate. Another phosphate you can join adenosine diphosphate. Another adenosine triphosphate. So, she is holding at C5 position carbon the phosphate group. And with another one, that will be C3. That will come later. Now it is free. Now, say it is free. This C5 is free. Then C3, this C3 will join with C4. Another C4 join with C5. Here, C5. So, here it will be like this. C5, C4, C3, C2, C1. So another sugar will remain joined with the previous nucleotide at C5 position with the help of phosphate PO4. This is known as phosphodiester bond. So, it will stand over the head of this next nucleotide. And this C1 will hold another balloon, guanine. Clear? So, adenine, guanine, phosphodiester bond, glycosidic bond. another C3, then here it will be C5 again. Again, this C1 will hold another balloon. These two are purine, adenine, guanine, thymine, and likewise cytosine also. And here also C3 and this C3, C5, this will hold, PO4 will hold a bond. This bond will be known as phosphodiester bond. Phosphodiester bond. This is a phosphodiester bond and this is a glycosidic bond. Now it is clear. So, in this way, this will go. You see, in every classes, we used to hear one thing. This is C 5 prime, this is 3 prime. Why it is so? Here you see, this 5 prime is free. C5 is free. And here C3 is free. So it is 5 prime to 3 prime. So this is the one side of the nucleotide. And this adenine will join with some others. I am coming with this way. In this way, same thing. Now, adenine, guanine, thymine. And in this way, my data I have told you that in the C5 position, C3 and C5 will join. Here C1 will hold the nitrogenous bases. So here also C1 will hold the nitrogenous bases and the base pairing rule, we know that ATGC formula, adenine with thymine and guanine with cytosine. One purine, one pyrimidine, one purine, one pyrimidine. These two are with 3 hydrogen bonds, these are with bonds, and these are with 2 hydrogen bonds. So here adenine will mix with join with thymine with double bond and guanine with cytosine, thymine with adenine. Now, if you want to join this, so okay, let me will join because this base will remain attached with the help of glycosidic bond with the deoxyribose sugar. So it has C1 here, C2 here, C3 here, C4 here, C5 here. So it will remain with, join with C1. Will it be like this? No. C will join with, G will join with C. Whether C will join in this way? No. Because it has to join with C1. How we can make it? Simply, this is the main pertinent question, because if you want to put this formula here also, it will be in reverse direction. Say it will be reversed. How? Now it will make sense. That is C1, C2, C3, C4, this is C5. Clear? Now this C1 will join with thymine base, or it will hold the balloon here by glycoside, by beta-glycosidic bond. So it will join here. Likewise, it joins here. So the sugar will be reversed. In this way, if you write here also, you write in this way. So here, here. Here it will be C1, C2, C3, C4 and C5 here. You put this figure here, only then you can join. So it will be, the sugar will be in the reverse way. It will join. Now you see, the problem is that it will again join with another this is C1, C2, C3, C4 and in the head C5. Okay? So, here this C1 will join here. So, we will say the nitrogenous bases will remain joined with the sugar at C1 carbon position. At C1 carbon position. Here also C1 carbon position. You see, here this 5 end is free, and here what end is free? This 5 end, this will phosphodiester bond, again this will make phosphodiester bond with the other, this C3 is free. So this is known as 3' to 5' will last one will have C5 free, so this is 5'. We know that 5' to 3', 3' to 5', this is the main thing about 5' to 3'. So this is DNA. And this 4 nucleotide produces tetranucleotide. In the cell, we will get nucleotide 10,000 and more nucleotides are given. So this type of nucleotides are so long, it remains 10,000 to more than 10,000. So many nucleotides remain there and it remains in a helical way. What is that helical way? You have gone to this is the backbone phosphodiester bond. And here a little bit more will be required. Space is required of the staircase. So it will remain joined with A, T, G, C, T, A, C, G, like this. That is one part. These nitrogenous bases will be inside and sugar and phosphate will be the outside. This is sugar and phosphate bond. Say sugar and phosphate, sugar and phosphate, sugar and phosphate. In this way, they will prepare a railing on both sides. On both sides. Okay. Now, A will join with double bond T. T will A, G, C, C, G. T double bond A double bond T, C triple bond G, G triple bond C. So this will remain in the number one position, it will remain joined. This is phosphodiester bond and these are known as glycosidic bond. Clear? Now you see, you have gone to circus, because in circus the ladder is used is a flexible one. In circus, in trapeze, in the first opening ceremony, what you will see that there is a ladder of rope with having steps like this. It is tied in the above, and whenever the game starts, they open this and it flows here up to this. And with the help of this ladder, the beautiful girls as well as boys having good physique and body fitness, they climb. They climb and goes there and sit there in their specified position. Now, whenever this climb, you see this floats here and there because it is flexible. So it also it is a flexible. If you turn it clockwise here in this way, it will be here like this. This bond will be like this. So it will be here like this. This is the turn. Now, in book it is written, this distance is 2 angstrom unit or 2 nanometer, whatever it is, you see. And one step to another step is 3.4 angstrom unit or nanometer. Somewhere it is written nanometer, somewhere it is written angstrom unit. So 1, 2, 3, 4, 5, 6 like this. It has been seen that this rope take a full turn from here. It will take a full turn here to this. So it takes a full turn. This full turn will hold 10 base. 10 bases. 3.4 into 10 is equal to 34. And this turn up to this turn, in a single turn, it is 34 angstrom unit. This is the simple thing you should remind, that the helical structure or the phosphodiester bond take a full turn in every 34 angstrom unit, and the difference between the two base pair, the two base from this to this pair is 2 angstrom unit, and if a full term, it contains 10 bases. So whenever it takes a full term, this full term, this to this, it got 10 bases. 10 ladders are there. So this is somebody calls this is major group, this is minor group, Forget it is need not to be discussed. Minor group, major group, and this holds 10. So how many number of these stars are there? You can easily count that how many bases are there without counting 1, 2, 3, 4, 5. So if you get a term of say 360 term, 360 into 10 or 3600 into 10. So in this way these are counted. So this DNA molecule is not 1, 2, 3, 4 tetranucleotide. It is linear polymers of pyrimidine and purine nucleotide. So it is a linear molecule of DNA is a linear molecule of purine and pyrimidine nucleotide joins one with another with the help of phosphodiester bond, and the bases remain joined with the sugar at C1 position with the help of glycosidic bond. And the two bases remain joined with each other by HY, DRO, GEN hydrogen bonds. Clear? So up to this, it is DNA. And on the next day, we'll simply cut it here. We'll cut with the help of helicase and we'll replicate one another with the help of DNA replication. That is producing a replica of this. That is very simple. On the next episode, we'll do that. Till then, thank you very much.